So we combine different tools, taking advantage of the model system that we're working with. And so one of them is actually what we call virtual reality, which is essentially we put the fly to play video games. We immerse the fly in the same way that we're immersed when we're trying to play any video game in a world. And we characterize or construct different worlds depending on what exactly we want to ask. Let's imagine that we're moving our heads and our eyes to the right. What we see is that the world is moving to the left. We know that the world is static because the brain knows that we're moving. And so we don't understand in the field of neuroscience how exactly is that the brain gets to represent this internal information. So what we do in the lab is basically trying to answer that question. The advantage of putting the fly in this virtual reality world is that we can control the world very well, so we know exactly what kind of sensory information or what kind of a state of the world the animal is in. And we combine this with perturbations of different populations of neurons using the genetic tools of the fly, and that will indicate us what neurons and what regions in the brain of the fly are involved in controlling the interaction of the animal with this virtual world. Let's imagine that we're trying to walk on a straight line. We think of this as a very simple task, but in fact it's very difficult to do. And you will notice this immediately after you close your eyes and you try to maintain this trajectory or path in a straight line. After a couple of steps, your path is going to be deterring from a straight course. So what we had just recently found is that this is exactly the same for the fly. The fly depends a lot on visual information that is generated by her own movements to be able to maintain a very straight course. The second thing we have actually recently discovered is a population of neurons, a neural network that is actually processing this kind of visual information, and it's also able to control the movements of the fly. So we are now in a really great and unique position to be able to understand how these multiple kind of signals are combined in this network to generate these internal representations of the movements of the fly, and what's the consequence of that activity in controlling a straight course of the animal. Insects and humans are separated by millions of years of evolution, and yet the control of their locomotive system is remarkably similar. So our strong working hypothesis here is that what we can learn from this tiny fly will extract general principles that can be applied to other animals, including humans. So we are really grateful to the ERC for our starting grant because it allows us to take on this very ambitious project in a slightly different and very creative level. Having the flexibility that the ERC gives you allows to recruit very talented and diverse people from all around the world, and that creates an environment that nurtures creativity and really thinking about what kind of experiments and what is the proper set of experiments for the questions you're trying to address. Not only it allows us to get the proper technology to run the experiments we are proposing to do, but it also allows us to develop technology. In the particular context of our motor control system, we might actually learn how to develop technology that will assist and facilitate conditions in which motor control is actually affected.